Okay, my name is Matthew Wokanski. I am teaching two of the pathology sections for this term, and I'm doing this lecture for Annabelle Serrano as well. So in this lecture right now, we are going to talk about something that's very timely. We're going to be talking about infection and the things that actually cause infections, and some of the aspects of it. In particular, we're going to talk about the learning objectives for this specific lecture that we're going to do right now. We're going to talk about different types of pathological organisms. We're going to talk about issues of susceptibility. We are going to talk about something called virulence of the organism as well. And then we're going to talk about two measures, very simple stuff. We're going to delve into a little bit, very little epidemiology here, but something that is very timely in today's age, some basic math that some people just can't get around their heads. And I'll get to that towards the end of the lecture. And so when we talk about an infection, what are we talking about? We are talking about some sort of invasion in the body by some sort of pathogen. The word pathogen basically means a microorganism that causes disease. Okay, specifically causes disease in humans. So if we say a human pathogen, we're talking about something that causes disease. There are some bacteria that cause disease in other animals, but not in us. So they would be a specific pathogen for a certain species. We see that there are specific pathogens for us. Everybody, when they think of pathogen, usually thinks of bacteria and viruses, which we will talk about. But it's important to note that that word pathogen is very specific to us because if, for example, when we go to talk about bacteria, we will find that there are hundreds of thousands of different type of bacteria that you come in contact on a regular basis that are not bad at all to you. Actually, some of them make the best tasting yogurt. So we can see that specifically in yogurt doesn't kill you, although some people who dislike health food might think so. But anything that it causes an illness or causes some sort of disease, we call a pathogen. And if you were to look zoologically, meaning looking at all the different types of viruses and bacteria that are out there, we're talking about a very small number that actually cause disease in humans. But there's enough, and they're common enough that they cause diseases. So this results in symptoms being developed in response to the invasion. We can have infections that are either localized to a small area. You can have infections in skin, in joints, under certain tissues, or it could go systemic, which means that it actually gets into that pathway that you're going to talk about in anatomy this week, which is the bloodstream, and it goes through the bloodstream. And if it goes through the bloodstream, then it gets everywhere within the body. And when we talk about infectious agents, we're talking about, when we said pathogen, it's something that has the potential to do harm or will do harm if it gets into a human being. Example of some pathogens that we're going to talk about, bacteria, as we said, is what I said on the previous slide. Also viruses, the most interesting one right now is our good friend the coronavirus. And we have also other pathogens that can cause illnesses are fungi and also parasites. Now, some conceptual notes about some infectious agents. Let's look at these things. As I said, there are these four things that we talked about on the previous slide. We mentioned there are structural differences with each one of them. And with those structural differences, we are going to see that those structural, meaning how a bacteria is made up, how a virus is made up, basically will dictate or determine how a patient can get it and how the pathogen can then cause harm, okay? And how then we would actually treat it. So we're going to start in pathology. This is why pathology is very important, because by knowing the pathology on how something infects you, you can know how to defeat it with certain medications, okay? And suffice it to say, if Donald Trump knew anything about biology, he wouldn't be making excuse my language, he would not be making statements like, oh, let's just ingest some Javex to see whether or not we can get rid of a virus in the body. Yes, I'm going to make a few off-color jokes in this slide about Donald Trump. If you are a Trump forever sort of person, you just might want to skip these slides. So when we talk about infectious agents, we're talking about some microorganisms. Now, we talked about I mentioned four that everybody knows, or two that everybody knows, bacteria and viruses, mentioned fungi, mentioned and mentioned parasites. There's actually something else that has been around that is sort of under control right now, but we still worry about it. It's something called prions. And prions, what the hell is that? You might have never heard of prions. There hasn't been a big resurgent in North America in the last 10 to 20 years. But at a time, if you were to put this term mad cow disease into Google, you will get something that's called Creutzfeldt-Jakob disease, which is not actually what we think is the norm about what we think about viruses and bacteria, fungi and parasites. And what is very different about this is the genomic material that we're looking at. Look at these things down here. Look at viruses, bacteria, fungi, and parasites. DNA, 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 DNA. And in viruses, we know that some viruses can use RNA to cause illnesses within patients. Look at this one up here, prions, protein particles. Protein particles? Yeah, for some reason, if you remember back to your biology, your basic biology, DNA is the nuclear material that creates something called RNA, and then RNA then translates to create proteins. So we know that, as we're going to see in a couple of slides, that some RNAs can, and viruses, once they get into the body, can reverse transcript themselves. So going from RNA or DNA to RNA to a protein, that process is called transcription. And usually we go from RNA or DNA to RNA to a protein. Well, we know in some viruses, they have an enzyme called reverse transcriptase, and what it can do is it can take that RNA and create DNA that can then be infected into your cells. Well, with these protein particles, we're sort of guessing right now because we're not sure exactly how they work, but some of these protein particles might be able to reverse transcribe themselves from protein to RNA to DNA, or they do something else wild and wonderful within our cells. But there's only pretty much one disease that we know of that causes this, so we're kind of happy that we haven't found any others. But the way the world is going, who knows? The one thing so the genomic material is very specific about and we see it bacteria, fungi, and parasites. Only DNA is their primary genomic material. We see that in replication, we see that, oh, look at this. Prions and viruses are host dependent. That means that a virus cannot replicate on its own. If you sneeze on a desk and you have COVID and you sneeze out exactly one COVID virus onto your desk, that one COVID virus will stay exactly the way it is for a period of time until it basically breaks down. It will not be able to replicate on its own. But if you were to sneeze into a cup of coffee, which has my favorite is usually, Tim Hortons double or triple cream, throw a little sugar in there, guess what's going to happen? Something outside of the body. If you stick bacteria or fungi in there, they might actually replicate within that cup of coffee. Don't believe me? Try it. Do a little backwash in a cup of coffee that has at least cream in it. Stick it on your desk and wait and check it out next week to next Friday. You will have some scum floating atop it. Well, that's either a fungus or that is either fungus or bacteria. You want to do something even less disgusting, just leave a loaf of bread out for a couple of weeks in a plastic bag. That green stuff, well, that's fungi, okay? And it grew all by itself outside of your body, okay? So we see that these things here they can self-replicate on their own. These two things up here need to be inside of a host to self-replicate, okay? Otherwise, they just disintegrate on their own. So here are the examples that we have from that. We have, I said, mad cow disease for prions is the only one we know of. Viruses, we have hepatitis, H1N1 cold. We could put COVID in there right now. bacteria, E. coli, strep, staphylococcus, fungi. The most common one is athlete's foot. Parasites, we see lice and tapeworms. Now, as I said with those prions, we'll talk a little bit about those in a little bit more detail. As we said, they are protein particles. They lack any real genomic material. They are pretty much protein particles, and they specifically happen in the bovine. That means basically means cow areas, and we call it bovine spongiform, and it causes something called bovine spongiform encephalopathy or mad cow disease. And the only problem with this is you're thinking, oh, who cares if a bunch of cows die? Well, the problem with that is if you ingest something that has mad cow disease, there's a possibility that you can get it as well. So it is transmissible from cows to humans. So if you eat the meat of a cow that is infected, you could get it. And it's transmissible neurodegenerative disease. And basically what it does is it starts to destroy your nervous system. And we end up getting something called ataxia, dementia, and basically death that can occur over months to years. We are unable to treat this because we're not sure exactly how it works. We know what it is, but we don't know what it does inside your cells or inside your body exactly. So, because we don't know how to treat it, we don't know how to stop it. But luckily this is the interesting part that I'm just going to throw in here. Luckily, the way that we've been able to get mad cow disease under control has been good old-fashioned isolation, disinfecting, okay, and distancing. So how we got mad cow disease under control was if we found mad cow disease in any herd of cows, basically that cow herd destroyed. We got rid of them. We did not sell them. We did not do anything with them. It's just to get rid of them all. Anybody who had been on a farm who leaves a farm would then would have to have their shoes disinfected, or they would go through certain procedures, cleaning procedures, and that's how we actually got mad cow disease under control in North America. Okay, there are these outbreaks, but there are controls in place just based on good old-fashioned isolation procedures that could stop something as nasty that we don't even have a cure for. I wonder where I'm leading towards on that idea. Viruses, generally not considered alive. I know if you go online right now, one of you does this every semester. Somebody is really good in biology and will go, well, there's an ongoing debate on whether or not viruses are considered alive or not and everything. I really don't care. Because you know what? That's for microbiologists to argue over beer and coffee, okay? Don't care. Not necessarily both at the same time, take your choice. But basically viruses, we will consider the most current statement from the group of from the international groups of biologists, which says that viruses are not alive because, think back to the previous slide, 2 slides ago, we said that viruses could not replicate on their own. So right now, the standard for considering something alive is whether or not it can replicate on its own or whether or not it needs a host. And they do not have all the cellular material that a bacteria does, so that's why it can't replicate on its own, or it doesn't have any potential to replicate on its own. So we essentially call viruses genes in a box, where you basically have some sort of a shell and then the DNA or RNA material inside, and that's it. If you think back to your basic biology, which you should all know, how a cell works, there's a hell of a lot more within a cell than just a membrane or a box and some DNA material. There's ribosomes, there's mitochondria, there's enzymes, there's endoplasmic reticulums, all those wonderful little things you remember about a cell. Well, those sort of things are in bacteria as well, to a certain degree, and that allows them to replicate on their own because they all have the machinery within them to replicate on their own. Viruses don't have all that junk. They just have some DNA material sitting in a capsule. Okay. Now there's 2 types we have that we talk about membranes. We have what's called a naked virus, and a naked virus basically has what we call a protein capsid around it here. So it has a bit of nucleic acid with a protein coat, which is called a capsid, and then sometimes that capsid could be enveloped in another covering, which is called a membrane envelope, and sometimes it's a phospholipid membrane, and we call this a coated virus, and we call this a naked virus. Okay, what does that mean? Well, unfortunately, that means that some of these viruses here in this coating here, they have receptors which make them very easily to change their, to mutate, and very easily can sometimes link onto humans. So sometimes these membrane encoded viruses can be much more dangerous to humans than a naked one. One sort of example here of a membrane encoded virus that is quite common is HIV. This is a structural model of HIV. So here we have, see these little white balls in here? There are these little round balls. This is the capsid inside. There is the RNA for HIV inside, has something called a reverse transcriptase enzyme that goes along with it, and then has a big membrane coating that goes around it, and that membrane coated has these little glycoprotein little stubs that come out of it, which, like certain cells within your body, and in particular when we're talking about HIV, these little glycoproteins here will lock onto cells within your immune system. So they're very specific and they like to mutate, which is what made HIV such a pain in the butt to get under control for a number of years. It was always changing this outside coat for a number of years. So that brings us, you're saying, why did you bring up HIV? Well, because there are certain viruses, one in particular that's going to come up in pathology again when we talk about neurodegenerative disorders. There are certain viruses that sometimes can stay latent within your cells, which means you get infected with them and then they just sort of hang out. And then they wait for a very opportune time when your immune system drops to sort of make themselves known. And this is a very common thing in the body. Some latent viruses are basically the herpes virus. Okay. You got a cold sore when you were a teenager, and then all of a sudden, several years later, you get another cold sore, and you've been single for the last year. Why is that? Well, all the circumstances worked out so that little latent herpes virus that has been basically sitting in your cells basically decided to rear its ugly head. Anybody who is listening to this lecture who did not get the chickenpox vaccination, who actually got chickenpox, guess what? Chickenpox is in you to stay. It's there. So that means as you get older and changes happen to your immune system, this is why people get outbreaks of shingles. Shingles is basically chickenpox virus that you got as a kid. Now, during the right set of circumstances where your body is a little bit more susceptible, boom, has an outbreak. Okay, so if you had chickenpox as a kid, like me, you still got it. Okay. The influenza virus, H1N1, is just a straightforward virus. And having said this right here, latent viruses, the HIV virus can go into latent phases as well. Influenza viruses are the most common one. We saw a very severe one called SARS in 2003 that we were able to get under control, not with a vaccine, again, with good PPE and good infection control, we were able to get SARS under control. HIV, as we talked about, was in the 1980s called AIDS. There is a thing here. HIV is the actual virus. Because it can go latent in your cells, you can be infected without showing any symptoms, okay? And then when you start to show symptoms, you end up getting the description of the symptoms you get as the HIV virus goes active is called AIDS. That's why people say they're living with HIV, but they do not have AIDS. Why? Because the HIV virus, under current medication protocols, can be kept basically sometimes latent forever. Okay, we've gotten to that stage where just taking your constant HIV medication, you can keep the HIV virus down to almost no action whatsoever. If they go off the medication, there's a potential the HIV virus replicates, goes active, and then destroys your immune system, and then you would have acquired immune deficiency syndrome. HIV is the virus that lives inside you. It's almost like up here, like a shingles virus that sits around waiting for the right time. The only problem is HIV, when it rears its ugly heads and becomes active, it can kill you. As I said, HIV is a retrovirus. It's a virus that has a RNA genome in it. Once that RNA genome gets into a cell, then it has a virus, or as it has an enzyme that does the following thing, has an enzyme that basically reverse transcribes it into a piece of DNA and then inserts it into your DNA in your cell. So it's literally like it's sticking a piece of its foreign DNA in your DNA. And then it waits until the right time to become active and destroy your immune system. It's not very nice. Another one that we had was MERS, and what we call COVID in 2012. MERS is still around. Middle Eastern I think it's called Middle Eastern retrosyncytial virus. This one had what we called a higher level of mortality to it, which was a concern, and we've sort of gotten that one under control as well. Of course, we can put down here as well COVID, and we'll talk about COVID in a little bit when we get towards the end of the lecture. Bacteria. Bacteria are not viruses in a boxes. They have all the junk that they need to live. As I said, you don't believe me? Take a cup of coffee, do a little back switch in it, leave it on your desk covered for a good week. You will see stuff growing. That's the bacteria that was in your mouth. Okay, so they do not require living tissue to survive. They just basically need sugar, warm, moist environment for most of the times. And bacteria have, although they do like being in your body, they don't necessarily need it. Bacteria have all the cellular mechanisms of life, and they fall under a category called prokaryotes, a prokaryote type of cell. Um, some bacteria are called pathogen causing disease, but as I said, most bacteria at the beginning of the lecture, most bacteria on our bodies are benign or beneficial. You have bacteria in your gut right now digesting food for you. You go to Yellowstone National Park. The reason why the stone is yellow is because it's bacteria. You know, there's bacteria everywhere, but it's not necessarily going to cause you illness. If it doesn't cause you illness, and it's benign, then it's not necessarily a pathogen for you. Bacteria. Well, bacteria are very adaptable. Okay, we are seeing that each species has a defined set of growth parameters, and usually the ones that cause damage or issues in humans are the ones that like the same temperature, atmosphere, and environment of your body. Okay, now people think that, oh, you know, bacteria can only live at a certain temperature. Only the ones that cause problems to you. There are actually bacteria that live in very cold environments. There's also bacteria that live in very hot environments, and also bacteria that live in environments that don't have any oxygen in it, and actually there are some bacteria that live on a sulfur diet. If you go to, there's actually bacteria that has been found at the bottom of the ocean by next to volcanic vents. Basically, they're vents where hot water that move around a volcano gets spewed out, and the water is like over 100 degrees Celsius and higher, and there's bacteria living down there in that environment. There's some bacteria that has been found in Death Valley, in some of the hottest areas of Arizona where there are sulfur springs. And they're finding bacteria there too, living there. Does this bacteria like you? No. If it got into you, it wouldn't cause any damage, most likely. But we see that bacteria are very adaptable. They can live at extreme temperatures and atmospheres. The ones that we're most concerned about are the ones that like our parameters for temperature, atmosphere, and environment. And we can see an example when we talk about us. We're talking about aerobes, one that require oxygen for growth and like a certain temperature, well, probably like your lung tissue. Anaerobes can't survive in an oxygen environment, but we can see some of these can live in certain areas of your body as well. Now, in particular, you're going to have to know a couple of very simple things about bacterial shapes. And the reason being is because you're going to see certain things popping up on lab reports for certain things what we call cultures, and you need to be able to recognize that if something comes up in a lab report and it says, first results from a patient's bacterial test is cocci in clusters. You need to know that that means that that's a positive test, and there's something there that needs to be addressed. Okay, so you need to know that we have certain bacterial shapes, and some of these bacteria will respond to certain antibiotics that you'll learn about in semester 3. So this is why we give you a little smattering of it in semester 2. So bacterial shapes, they can come in round spherical shapes, and we call them cocci, and they occur sometimes in chains or clusters. And primarily, that is why you've heard of streptococci. Well, streptococci, they're spherical shape. We have ones that are rod-shaped, okay, which are bacilli, and they basically are thread-like or line up in threads. We have some that are spiral as well, that look like a corkscrew, and they are short and rigid. And there are some longer ones that are called spherochettes, and they look like this. This is what they look like under a microscope. You have the cocci in chains or clusters. You have the bacilli that like to line up from end to end, and you have a spherochette over here. And this is another one. Here's just one looking at one in a cluster. This one has a little bit more of a cluster as well. And here's another image of it as well, of the spherochettes. Now the bacteria, there is something in particular about bacteria that won't mean too much for you right now, but in semester 3, when you're learning about antibiotics, if you remember this, big time, okay? The one thing about this that we're going to talk about is that it's very important to how nasty some of these bacteria are, okay? Some pathogen bacteria can be nastier than others. Now, nearly all prokaryote bacteria have something that is different than the cells in your body are classified as eukaryotic cells. They're eukaryotic cells because they lack a cell wall. Bacteria have this extra thing called a cell wall. Provides, which this is what makes bacteria so resilient. It provides physical protection for them. It protects them against changing moisture conditions around them, very dry, very wet. And the thing that is inside these cell walls is something called peptidoglycan. You do not have peptidoglycan in your body. So that's a big difference between you and a prokaryote cell or a bacteria cell, is you do not have peptidoglycans in it. So when we go and see whether or not, if we see something under a microscope and we want to see whether or not it's a eukaryotic or prokaryotic cell, prokaryotic meaning bacteria, what we do is we have certain stains that are only attracted to peptidoglycans. So this is great. And this allows us, or if we're identifying a bacteria, we can use these stains on it. Eukaryotic cells will not take up any of the stain. And we will see that if we use a certain stain, and it's called a Gram stain. I think it's because it was named after the guy. Quite honestly, I've forgotten. But Gram stain, if we use this Gram stain, which peptidoglycans pick up very quickly, we see that we will have what's called a Gram positive or a Gram negative stain, which means Gram positive, we see that a lot of the stain is picked up and it turns really, really pink. In a Gram negative stain, we see a bacteria that has less peptidoglycan in its outer shell and doesn't pick up as much stain. Okay, we find that we have Gram negative, Gram negative bacteria that don't pick up a lot of this Gram stain, are usually more complex in their structure and are more likely to cause death. They have a higher death rate, these things. If you get a Gram negative bacteria, you're in big trouble. You better get some antibiotics real quick. If you have a Gram positive one, well, we got a ton of antibiotics that are good at going after Gram positives, and we'll explain why in the next couple of slides. So as we said, just as a very quick one here, we see this cell wall. We have is this very thick cell wall that goes around the bacteria like this. And the funny thing about inside of a bacteria is the other thing that's different between a eukaryotic cell and a prokaryotic cell, not only the cell wall, but what's missing here in the middle here. This is all DNA material. And this is all the machinery of the bacteria cell. So what does that mean? This thing doesn't have a nucleus a lot of bacteria just have their DNA floating all over the place. Okay, so that is something as well that differentiates you from a bacteria cell. Okay, and here's where we're going to talk about the differences in a gram-positive and a gram-negative cell. Down here we have a gram-negative cell for your orientation. Up here at the top, this is the outside of a bacteria. This is the outside of the bacteria for this little piece here. These are cutaways of a gram-positive bacteria cell walls, and this is a cutaway of a gram-negative bacteria cell wall. And this would be the outside over here. This would be the outside over here. If we were like trying to get into the bacteria here, we would go through these two layers of peptidoglycans and then a membrane before we would get into the cell. Over here, if we wanted to try and get into this bacteria, which has this sort of outside structure, we would have to go through a cell membrane, one layer of peptidoglycans, another membrane before we could get into the cell. Okay? And just having explained that right here, where you have a membrane, thin layer of peptidoglycan, and then another membrane, we say this is a more complex outer structure. We look down here at this gram-positive structure right here, and we see that we have two layers of peptidoglycan stacked on top of each other, and one layer of membrane. And the peptidoglycans are free and clear to interact with the outside environment. Okay, this is a much simpler structure. Okay, I consider this as two types of sandwiches. A gram-negative structure has a slice of bread, one little slice of bologna on the inside, and then another slice of bread on top. And that is more complex. Okay. Down here, we just have one slice of bread, and it's like going to Schwartz's Deli in Montreal. We just load a whole load of meat on top of it and forget the other slice of bread. Now, you're going, okay, why is he making this example? Because remember what we said on the previous slide. Okay, we throw a stain in that then is picked up by peptidoglycans. In a gram-positive stain, we throw the stain around the outside of the bacteria out here, and look, it very easily gets picked up by the peptidoglycans. The peptidoglycans are in close proximity to the stain. It gets absorbed through it. You've got two layers here. You get a nice bright color. Over here, in a gram-negative bacteria, you throw in a stain. Well, the stain's got to try and get through this membrane over here, and even if it does, it's only going to get picked up by one layer of peptidoglycans, so it's not going to dye itself as well, if not at all. Okay, so we say that this is more complex. It does not pick up stain because there is a barrier for it between picking up stain, which is another lipid membrane, as opposed to a gram-positive down here. Now you're going, who cares? Well, I did say that these ones can be more deadly. And the reason being is because they're protected by another membrane up here. Oddly enough, the thing that we use one of the most common antibiotics that we use, guess what structure it destroys? Peptidoglycans. You destroy the peptidoglycans, destroy the cell wall, which a majority of our antibiotics that you're going to learn about in semester 3, you have an antibiotic that likes to go after peptidoglycans and destroy it. You basically will cause the bacteria to basically implode, explode, collapse if you can destroy it. Well, if we have trouble getting stain to these peptidoglycans, you have trouble getting those common antibiotics in here, this thing becomes more resilient. Okay. Down here, this thing right here, all I have to do is throw a antibiotic in here that likes to destroy peptidoglycans, and Bob's your uncle, and we can destroy that bacteria. So that's why it's very important when we see a gram-positive cell wall, we know that we got tons of antibiotics to throw at this thing. If we see a gram-negative one, we know we got to up our game in our antibiotics and use something different. Again, I'm not going to go into the different types of antibiotics as much, just to give you a brief explanation for semester 3, why you need to know these criteria here. Now, fungi, let's go on to fungi. Fungi are basically, they are actually more like eukaryotic cells. Okay, they have a cell wall, yes, they do, but they also have a nuclei, which is different than bacteria. They are absorptive heterotrophic eukaryotes. What they basically do is when a fungi gets in contact with something, what it does is it secretes enzymes to whatever they're in contact with, and basically causes that substance to dissolve, and then they then absorb the nutrients or the byproducts of whatever they're dissolving. And there are two basic groups. We have molds and we have yeasts. And we all know that there are yeast infections in populations. And these yeasts can be, most common are sexually produced yeast infections. And some of them are, and yeast can reproduce by themselves asexually, which basically means they don't need any extra DNA from anything else. So whereas bacteria do, and basically they can reproduce asexually, and the most common ones are thrush, which you can get in your mouth. There are vaginal yeast infections. There are yeast infections that you can get under skin, on certain areas of skin, under your armpits where there are skin folds. Fungi are fun. The other most common one too is athlete's foot. And this is what fungi, yeast, and mold sort of look like. Okay, they're not fun. They can cause nasty little problems. The one thing that is very particular about fungi and molds and yeast is the way to tell them between some other type of infections is most of the times when we get a colony of them or a grouping of them, they are very smooth, or they can appear as smooth, waxy, or have a creamy texture to it. And we sometimes again see these infections in the inguinal areas around the groin areas. And sometimes they will also produce white cottony or powdery colonies, right? So if they look like this or have these sort of aspects, it's more than likely a fungal infections. And again, they do have a rigid cell wall that are not as susceptible to any penicillin-like antibiotics. But the thing that makes them eukaryotic cells is they have a nucleus, okay, which is similar to you. So believe it or not, you have more in common with a fungus than you do a bacteria. Interesting. And mushrooms are fungi too, so you're more like a fungi than you are like a bacteria. And there are only really 50 species of fungi that are parasitic on animals. And when you have a fungal infection, we call it a mycosis. Okay, and some of these ones are pretty common. We have ringworm because it appears like a circular red area on the skin. Athlete's foot, also caused by a ringworm fungus. Vaginal and yeast infections. If you hear that little noise in the background, that's my new puppy beating up on my older puppy. Some fungi also provide antibiotics for ourselves. You wonder where we get some. So this is one of my things. I've said it and all of you have, most of you, or 90% of you should have um, pharmacology with me this semester. And I have said in one of my lectures, we get most of our drugs from nature. Well, here is one of the most common nature drugs right away that we actually got. We have penicillin. Penicillin is the enzyme that is secreted by a fungus, and that fungus specifically secretes it to destroy peptidoglycan so that it could absorb the contents of beat up, broken down bacteria. Huh, how did we discover that? Well, this is something called a petri dish. Okay, it is the most common way for us to, what we say, culture. It's the most common way for us to culture bacteria so that we can actually grow a whole bunch of it and then look under it under a microscope. Still, the old-fashioned way is still very common. So what happened is one day somebody, and this uh, this tray right here, okay, or petri dish, basically has some, it has some nice sugar in it. It has a nice little, it has some fluid, some sugar, some other sort of things that bacteria love, and then it gets a little bit of moisture in it, and then we put it into a incubator at a bright temperature, and you try and grow bacteria in it. That's pretty much how it works. And what ended up happening in somebody's lab one day, they were trying to culture a bacteria, which this sort of wavy line right here is a bacteria growth. And oddly enough, some penicillin mold got on it. Maybe the guy was eating a moldy piece of bread when he was playing around with his petri dish, and some of that mold dropped onto the petri dish. Well, the penicillin fungi started to grow. And what we saw is where the penicillin grew, look at this, the bacteria will only come so far to the penicillin mold. And if this penicillin mold grows even bigger, what we will see is this zone of inhibited growth, or where the bacteria will get killed off and it won't grow any further. Well, what's happening is they found out that the penicillin mold here is releasing digestive enzymes that like to digest peptidoglycans. So some genius went, hey, I wonder if we can make in the lab, the same sort of junk that is being made by this penicillin mold. And they can. And this is where we came up with penicillin-based drugs. So it came from nature. So the next time you look at a loaf of bread that's going moldy, say, hey, that's where my antibiotics are coming from. Don't eat it, okay? You'll make yourself sick. But I'm just saying that's basically where penicillin came from mold that's probably growing off of bread or something like that. So very, very, very useful to know how we can destroy bacteria based on this. And here's one of the things. We actually use fungi to beat up on bacteria. Now, parasites are a little bit different. Parasites, again, they do need a living host to complete their life cycle, but they can still, as part of their life cycle, reproduce outside of the host. We will see that there are certain things called protozoa. These are basically unicellular eukaryotes, and basically we have malaria, dysentery. These are things that are very common, and they cause illnesses within us. We have a thing called helminths, which are basically tapeworms. And yes, we still see tapeworms in this day and age. I work at Sick Kids. I'm still working there part-time while I work full-time here. I've been there for over 23 years, and every year I get at least 10 people who say, you know, we found this in our kids. Our kid had this spaghetti strand that was coming out, out their butt. So we brought it in a plastic bag for you. We think it's a tapeworm. Yeah, that's a tapeworm. Okay, so kids get tapeworms. Why? Well, they're sticking their hand in the dirt and then sticking their hands in their mouth, and they pick up the eggs from a tapeworm. Arthropods are also vectors of certain infections. So arthropod basically it's an insect that transmits a virus or bacteria. The insect itself isn't the pathogen. It's just the taxi cab that brings it to you. So we see things like ticks that bring Lyme disease. We see mosquitoes that can bring malaria. This is interesting because you have mosquitoes which can pick up the protozoa that gives you malaria, and it can give it to you. So arthropods aren't necessarily the parasite itself, but what they do is they're like the taxi cab that bring it to you. And the other one is Zika virus is another one. We see I said Lyme disease and malaria, and Zika virus is the other most common one. And what's the other one? Oh, West Nile virus is also transmitted by mosquitoes as well. And we usually see that there's something called ectoparasites infect external body surfaces, causing local tissue damage. And this is lice, body lice, head lice. Okay, those are called ectoparasites. You need to know because those exist as well. Bed bugs, ectoparasites, And here's one of the most common parasites, Giardia. Since everybody has been, not traveling to the States and going to Algonquin Park, if you drank the water in Algonquin Park and then got diarrhea for a week, it's because of this little bummer here. It's called Giardia, and it basically gives you what we call beaver fever if you go to Algonquin Park. It likes to part of its life cycle is in some of the live animals like beavers and stuff like that, and it gets itself in the lakes and rivers of Algonquin Park, and if you drink the water without purifying it first, you could pick this little nasty thing up here and give yourself a bad case of gastrointestinal parasite. Here we have a parasite here. This is malaria playing around with your white blood cells. And what makes a person susceptible? So we've talked about all these different sort of things, and what makes you susceptible to having a certain illness? There are certain things that are um, that can either deal with either the host or deal with the actual pathogen itself. And you will know, and we're not going to go into this because you do this in nursing theory, I believe. There is the chain of infection, okay? So you have to be aware that we're talking about the actual pathogen that works into the chain of infection. And the chain of infection can be a number of different things. We can see, like we have sexual intercourse here, you go through animals, come back to you have needles, injury. There's a whole bunch of different ways that you can transmit and retransmit certain pathogens within your body. And the life cycle there's some areas where the life cycle actually then is actually outside of the body and then returns back into this cycle where it gets back in. So make sure that for nursing theory you understand that we are talking about the pathogens that you will learn about in the transmission of the chain of transmission when you go to nursing theory. So the host, now there's a couple of things we will talk about. So if we talk about in that chain of infection, if we have the host, okay, let's talk about that. Any organism is capable of supporting the physical growth requirements of another organism, okay? There is bacteria that like our body temperature, our level of sugar in our bloodstream, like our like that we're like certain like certain aspects of living on or in our body. And therefore that can make us susceptible. And it depends. What makes us susceptible sometimes is your level of resistance. Okay. Right now, you come in contact every day with viruses and bacteria all the time. Hate to tell you, you've probably inhaled in contact and come in contact with a whole bunch of things. But it's not enough to cause disease. Okay, because your immune system attacks it. It's only a small amount. Your body is able to overcome those bacteria or viruses and get rid of them, and you have no ill effects. Okay, so if your body's resistance or your body's resistant functions start to go downhill, then this is where you can fall ill to some very common ailments. The most common ailment I can say, remember a couple of slides back we talked about HIV and acquired immune deficiency syndrome? Well, AIDS basically destroys your body's immune system. And what ends up happening is people don't die of AIDS per se. The HIV virus destroys the person's immune system to the point where they have no resistance, and common bacteria that you would normally fight off end up killing you. Okay, one of the most common deaths, things that cause death for somebody who have full-blown AIDS, where their immune system is destroyed, is a pneumonia through something called pneumocystis carinii. Pneumocystis carinii, pretty much once you're past 2 or 3 months of age, your immune system just fights it off and kills it. Okay, and you do that through the majority of your life. But if you don't have any immune system, then your body cannot fight out this very common and routine bacteria which you come in contact with every day. So somebody's susceptibility, and in the case of somebody who has full-blown AIDS, they are very susceptible. And your susceptibility depends on that level of resistance that can be affected within the body. And as we know, we're not going to talk too much about it, but that chain of infection. So what would make a pathogen more more of an issue in the chain of infection is if somebody has, oh my God, where have we heard these terms before? Your general health status. So if you're generally in good health, then you're going to have a higher level of resistance. If you have a pre-existing condition, where we heard that before in the news, so where we see COVID is hitting a lot of people, are those people with pre-existing conditions, people with type 2 diabetes, people with underlying pneumonias, people with tuberculosis. So pre-existing conditions, we see that the level of resistance that a person can have is lower. And this is where we see that for COVID-19, we see the death rate is a lot higher there. We can see that certain age, if you're very, very old or the very, very young, that can have an effect on your immune system as well. If you're very young, you haven't been exposed to a lot, and if you're very old, well, things are just not working the way you want them to. We can see that there is some differences as well. There are some meaning biological sex differences for some people, where females or males are more susceptible based on the structure of their body. Okay, women tend to get a lot of yeast infections in the inguinal area, vaginal yeast infections. Men necessarily do not. Men are more susceptible to getting fungal infections if they're uncircumcised. Women, not so much so. Okay, and that's due to basic sexual or basic biological differences of the sexes. So it also depends on, and sometimes it also depends on the virulence of the infection agents. You can have all these things, you can be in good health, no pre-existing conditions, young and healthy, and da 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 da. But if you have a virus that has some issue that have some aspects out of what makes it very virulent, okay, it can even overcome somebody who has a good resistance. And what are some of those virulence factors? There are 4 that we're going to talk about over the next couple of pages here. First of all, we're going to talk about some pathogens that produce toxins, specifically ones that produce what we call endotoxins and exotoxins. One of these is much worse than the other, and it should be obvious when you read it. An endotoxin is a lipopolysaccharide, okay, in the cell wall of a gram-negative bacteria that attaches to a binding protein in a leukocyte cell. Check out your anatomy this week or next week when Jerry's group gets to it. Leukocytes are part of your immune system. So if we have a bacteria or we have a pathogen that releases a toxin that goes after your immune system, that's not good. Okay, that's destroying your immune system. So this one can alter immune response and make you even more susceptible. Okay, not good. I don't want an endotoxin. Exotoxin, bad, not as bad as an endotoxin. And look, look at gram-negative bacteria. Oh crap. Not only is it harder to kill with bacteria, go back a few slides, not only are those gram-negative bacteria harder to kill with antibiotics, but also they are more likely to release endotoxins, which go after your immune system. I wonder if something like that will show up on a test question. Just saying. Anyway, endotoxins or exotoxins here are proteins that are released from bacteria during growth. They enzymatically inactivate or modify some key cell functions, so they only go after the cell that they're kind of attached to. They can lead to cell death or dysfunction as well. These ones don't tend to go after your immune system, just the cells surrounding them. Some examples are botulism, tetanus, and cholera. Ugh. Now, the second sort of virulence factors, that's why there's a number 2 here. The first set of virulence factors were the toxins, endo and exo on the previous slide. The second virulent factor that a excuse me, that a pathogen can have is adhesion factors. So some pathogens are able to attach themselves to host cells and therefore colonize. And they have certain receptors that can come up and attach onto certain cells. That makes them very hard to remove. Sometimes they form a slime layer with anchors that then also protect themselves. We are going to see this when we talk about tuberculosis and respiratory issues in the next couple of weeks. Big deal here, okay? Tuberculosis is nasty because it has adhesion factors and it creates a slime layer as well. It coats itself. Pain in the butt, okay? Let's go on to the next virulence factor. The third and fourth virulence factors of some pathogens are what we call evasive factors and invasive factors. Invasive factors are mostly enzymes that are produced by infectious agents that help penetration into a certain group of tissues. So you have a bacteria that gets itself onto your lung tissue that produces some enzyme that allows it to penetrate deep into your tissues, or one that allows it to penetrate deep into your bone tissues. That's not nice. That's a kind of nasty sort of little thing. Or they have some invasive factors where what they do is they release some certain enzyme or chemicals around them that prevent the body from destroying them, almost like a little protective zone around them as well. And as you see here, the effect of pathogen invasive factors and toxins combined with antimicrobial and inflammatory substances released by the host cells mediate tissue damage and the pathophysiology of a lot of diseases. So you end up seeing, what this is end up saying is, when you have these sort of things going on, we are going to see, when we talk about tuberculosis, we are going to see something where you have something that adheres to certain tissues, creates a slime layer, actually releases one of these invasive and evasive factors, and actually causes the immune system to go into overdrive and around it, and actually that immune system response ends up causing more damage as well than it does actually helping. So when we get into tuberculosis, this will make a lot of sense. You might even, when we talk about tuberculosis, come back to these definitions and look closely at these definitions, because they're going to make sense when we talk about tuberculosis in respiratory diseases. Okay, so this is new for this semester. There's about 6 slides here, but how do we gauge? Like, we've told you all these different things about how a pathogen can, work its way into the body and stuff like this and everything. But how do we know how really bad it is when you throw it into a population of people? Well, this is where we have to know a little bit of epidemiology. Not a lot. There's only 2 things we really want you to know in semester 3 or semester 2, and that is a little bit of epidemiology and 2 terms. We'd like to know if a pathogen gets into a community, and a community is basically, it could be the community of Toronto, or the community of Ontario, or the community of Canada, or the community of North America, whatever you may say that your community defined community is. We would like to know how easily it moves or it distributes itself within that population. That means we want to know how easy it gets spread, and how far and how fast it gets spread. We also want to know on how, once it gets spread, how deadly is it once people get it. And there are 2 terms that do just that we look at. We look at the term morbidity, and this is the number of people in a given population. So right now, let's take a look at what we're doing right here in Canada. Right now, our borders are closed. Okay, nobody from the States can come in. Okay, none of us can go into the States for anything that's not non-essential. So right now, in the population, we have 2 separate populations in North America. We have the US and we have Canada. Canada's doing okay. Let's talk about the States. So if we look at the morbidity in the States, we will see that, given that closed population of the United States, there will be a number of people that will get the disease, and let's use COVID-19. There are a number of people that will get, the morbidity rate is very high for COVID-19. It means people will get the disease and it's spread very easily, okay? And these people can be, and getting the disease means you can also be symptomatic or asymptomatic. Symptomatic means, okay, I got a cough and a cold, I feel horrible, I got the disease. Or I'm asymptomatic, which means I'm really not feeling sick, but I got the swab and I got COVID. Okay, so that falls under morbidity, the number of people that got the disease or got the illness. Okay, if we're talking about COVID, we're talking about the number of people that have confirmed that they have COVID in their little bodies. Then we look at, and this number is a proportion of the whole population. So the way that we look at morbidity is, out of the entire population of the US, which is 328 million, the morbidity is the number of people out of that 328 million that got COVID-19. Okay, that is morbidity. You just got the disease. All right, and it is expressed as a percentage. All right, so we would say the morbidity is 11%. That means 11% of the population will get it. Okay, if nothing is done. If we look at the mortality, this is the number of people that got sick that then die from it. Okay, so if we say the mortality rate is 1%, if we say the mortality rate is less than 1%, that's that percentage here. The morbidity is the number of people out of the entire population you're looking at. Mortality is a number which is a percentage of these people here of morbidity that died. Okay, that needs to be very clear on what group how we're talking about this. And this is why I made this little diagram right here. Let's look at the population of the United States is $328 million, or 300 million. That's what I wish I would actually win in a lottery, $328 million. In this United States right now, we have 328 million people is their last population estimate as of 2019. So that's 3 out of 328 million people. Some of them will get sick. So a small portion of them will get sick with COVID-19. And then of this population here that got sick, some will die. Okay, can't make it any easier than that. Okay, so this number is a function of how many died in this number, and this number is how many got sick from the entire population that you're looking at. Okay, now what does that mean? What do the numbers mean? Well, we have to consider how really bad it is, and of this right here, I sort of make these little diagrams, and who's saying whether or not it's low, medium, or high? Well, this is me looking at the numbers, okay? So if you try and go find this online somewhere and say, well, is it low, medium, or high? I'm doing this based on morbidity and mortality numbers and sort of giving it a qualification in my head. So let's look at something that has pretty low morbidity, meaning there's like less than a fraction of a percent of people who get this anytime during the year in Canada or the United States. But if you got it, you without treatment, you're gonna die. Okay, that's something called bacterial meningitis, and we would consider that a pretty bad illness because, as I said, you have to sort of look at the combination here. If I have something that will kill you if you get it, even though there's a very small percentage of people that will get it, it's pretty bad because the people who get it are going to die. Not good. Okay, so we consider that a pretty bad illness. Okay, so bacterial meningitis is pretty bad if you get it. If we have something that has high morbidity but really low mortality, not a lot of people die from it, right? That basically means that are you going to have a bad outcome if you get sick? No, not really. The morbidity number is pretty much, not a lot of people die out of that morbidity number. That's not a really big concern. The common cold is one of those things. It does kill people, don't get me wrong. It usually kills people who have really bad underlying conditions. But it's really not a big nerve. There's no, people don't bat an eyelash about the common cold. If we look at something that has high morbidity and medium, and I would say medium to possibly high mortality, depending on who's talking about what they consider high mortality. I'll say it's definitely bigger than the common cold, so we'll call it medium, which is COVID. You now have a pandemic. Isn't that suck? Okay, now let's look at some other illnesses which have a very high morbidity and a very high mortality. Okay, measles and polio. But you know what we got in place for that? Vaccines. And if you don't think these things are bad, go onto Google, type in polio 1940s, 1950s. Anybody who got polio. Polio was, let's not even talk about polio, let's talk about measles. Extremely highly contagious, extremely high bad mortalities and morbidities for young children. Okay, so bad. That's why we have mandatory vaccinations. It's not an issue these days because people have been vaccinated, okay? Unless you're somebody who doesn't believe in vaccinations. Not going to get me started on this one and see what I think about that. I do not believe in natural vaccination because guess what? If you get naturally vaccinated, you're going to have a high mortality rate. Just saying, I had somebody once who came into the emergency room and said, "Oh, we don't believe in vaccinations. We're going to get our children naturally vaccinated." And I said, "Really? So your child hasn't been vaccinated for anything?" She goes, "No." It was a parent who said this, and it was a she. She said, "No, we're going to get our child and do all natural vaccinations." And I said, "Really?" I said, "You let me know how that polio goes. Because you get polio, you end up with basically neurological damage and sometimes death. You get polio, you don't live." I just said, "Yeah, tell me how that polio vaccination, that natural polio vaccination goes when you get it." So there are some things that are just, you'll want to get vaccinated for it. So there are some things, and because of these, we've gotten these things that have high mortality, high morbidity under control. This thing right now, we don't have under control. Let's talk a little bit more about that. So why specifically is COVID-19 a pandemic? Well, COVID-19 right now is left unchecked. Unlike the flu, which some geniuses have been saying the two are alike, they are not. It COVID-19 is a type of, don't get me wrong, COVID-19 structurally is a virus that is similar to a common cold virus. The only problem is it is much more highly contagious and it's this mutated cold virus has a much higher mortality rate as well. So if it's left unchecked with no precautions, it has a high morbidity rate and a higher mortality rate than what you would expect of a common cold or the flu. In other words, it spreads very easily through the normal community environment. And when I say normal community environment, I don't mean lockdown PPE wearing masks. Okay, I mean what we were doing back in February, which is just running around, going to parties, doing that, going into stores, standing in line, less than 6 meters apart from people. It is more deadly than the seasonal flu. Remember as well, though, for the seasonal flu, there is a vaccine for it. I'm going to point that out for a reason when we get to the next slide. Now, you may think by looking at the next slide that I'm going to show you that the numbers of the percentages are quite low. You're like, oh, you're talking like, really low percent. Like they're not even like 50%. Why are we so worried about it? But if you we're going to take an example and we're going to do some simple calculations, okay, and look at what the percentages turn into when we talk about number of people alive at the end of all this and see what it is. So let's do the math. And by the way, this is 1135 Rebecca math, okay? Even simpler than that. Here is what the projected, and let's do the math and compare COVID-19 next to the seasonal flu, which some genius suggested the two of them were like. So what does that mean? Well, COVID-19 has a morbidity rate, and this is taken from the most current rates in the United States, because I'm talking about the United States because they have big numbers that we can look at, okay? And because they're the best example of a whole country that screwed it up. So let's look at the morbidity rate. Right now, since I looked at some numbers through August, the highest rates of COVID morbidity in certain states were anywhere between 8.8 to 10% of the entire population. The flu virus is anywhere between 3 and 11%. And you'd say to me, hey, dude, look, the morbidity rate is pretty much the same. Come on, it's no bit worse than the flu. Actually, the flu's worse. It's got to 11%, dude. Who do I sound like? Sound like Donald. Anyway, so you would say that these are similar, but look down here. The mortality rate of those 8.8 to 10% people, 3 to 4% will die compared to the flu, which has only got 1% of these 3 to 11% will die. Let's plug in the math. Current population, 328 million people. Take 8.8% and 10% of 328 million, you get a morbidity of 28.64 to 32.8 million people left unchecked will get COVID-19. Well, that's not too bad. This is because like really, but take 3% of these numbers, and I just used 3%. I just used 3% of this, and and let's look at the numbers down here, as I said, and I'm going to be really generous here. Okay, I'm just going to use. I'm not going to use. I know it says 3 to 4%. I'm not even going to use 4%. I'm not even going to use 3.5%. Let's just take, and I'm not even going to use the upper limit. Let's say it's no bigger than 8.8%. Okay, let's err on the side of, let's be really conservative with these numbers. If I only use 3%, which this mortality rate is pretty much confirmed across the world here. If we use 3% and times it by that 8.8% of 328 million, we end up getting the following. So remember we said 28.864 million people will get COVID-19. 3% of that is 865,920 dead. So close to 1 million people, if left unchecked, if nothing was done, no social distancing, no nothing, zilch, zada, close to 1 million people would die within 1 year. And these rates are usually done within 1 calendar year. Okay? So 1 calendar year, you could have close to 1 million, like almost 900,000 people dead in the United States. That's a big number, okay? Now, in comparison with the flu, if we do the same sort of calculations, let's do 3 to 11%, and I'll do the big calculations. Let's go to the top limit for the flu here. And let's say that 3 to 11% of the population get infected. We have 9.84 million to over 36 million people get the flu every year. And we see that without doing anything, we see that at a 1% mortality rate, we could have 96,400 people dead. Not 900, not 300, not 806, like closer to 100,000, like an eighth of this. Okay, we have only about 100,000 people up to 300,000, 360,000 people dying. Okay, and you're like, well, that's not too bad. Now let's look at the following right now. Okay, now let's take one thing into consideration on the next slide. Keep this number in mind right here, and let's look at the next slide. The current death toll from the flu in the US and Canada is somewhere around 80,000 in the last couple of years. That's not even close to the 96,000 that we just said. And you're saying to me, oh, but dude, that's because your numbers are wrong. It's really not that bad. You know, it's being oversold by the Democrats. No. What do we have in place right now that we do? Well, we have yearly vaccinations for the flu. We have a vaccine. Okay, and so we end up getting a lower predicted mortality and morbidity because we vaccinate a whole bunch of people, and we make sure that we vaccinate our people who are most vulnerable. So we end up getting a death rate that is lower than what could be predicted if we did nothing at all. Right now, there is no vaccine for COVID-19. So potentially, if the United States just threw away all their masks, what's a good portion of them want to do, and just, all of them go back to work and do nothing and go back to school, you could get up to that. You could get close to, within 1 calendar year, get up to that 1 million mark. Okay, there are certain estimates right now that with the current trends and the current numbers, they could get up to at least 300,000 by the end of this year, and that's not even a full year. So what does this all mean? Well, it means that this guy never passed Math 1135, could never pass Rebecca's math, and probably didn't even pass grade 5 math. Having said that, you guys have a couple more lectures that you need to do and move on with, and this is the end of lecture number 1.